Inside every human body beneath skin, memory, language, and name, there is an older record moving in silence. It has no alphabet, no carved monument, no ruined temple standing in the desert wind. Yet it carries traces of hunger, fever, birth, migration, and survival from ages so deep that no witness could ever speak of them. A drop of blood seems small, almost ordinary, but within it lies one of the strangest inheritances of the human story, a set of invisible markers passed from parent to child, shaped across thousands of generations, and scattered unevenly across the world like the remains of an ancient map. Long before anyone called it type A, type B, type AB, or type O, blood was simply life itself. It darkened stone tools when hunters butchered animals on cold grasslands. It stained the hands of healers who could not see what made one wound close and another turn fatal. It flowed in childbirth beside fires, in battle beside city walls, in ritual places where people believed the boundary between the living and the dead could briefly open. For most of history, humans knew blood by sight, smell, and fear, but they did not know that one person's blood could differ from another's in ways hidden from the eye. That hidden difference mattered. It mattered when ancient diseases moved through crowded settlements. It mattered when small bands crossed deserts, forests, and frozen corridors, carrying not only tools and stories, but genes. It mattered when isolated populations grew for centuries with little outside contact. It mattered when farmers, herders, sailors, soldiers, captives, merchants, and refugees carried their blood markers across continents. What survives in the record suggests that blood types are not random labels. They're not simple signs of personality or destiny, as some popular traditions later imagined. They're biological fossils of a kind, living traces of pressures that once determined who endured, who suffered, and who left descendants behind. Before we go deeper, take a moment to like the video and subscribe if this is the kind of history and mystery you enjoy. The mystery begins with a quiet contradiction. All human blood looks essentially the same when it spills. It pools, red darkens as it dries, and seems to belong to one shared human condition. Yet at the microscopic level, red blood cells carry tiny surface structures like molecular flags. These markers can decide whether a transfusion saves a life or destroys it. They can shape risk during pregnancy. They may influence how certain pathogens attach to cells. They can reveal old patterns of ancestry, though never with the simplicity people sometimes wish for. Blood type is both deeply personal and far older than any individual life. The ABO system. The most famous of these blood group systems is only one part of a much larger world. Modern science recognizes many blood group systems, each with its own genetic history. But ABO became famous because it is so medically important. The difference between receiving compatible and incompatible blood can be the difference between recovery and catastrophe. Yet the same system that transformed medicine in the modern age was already ancient before medicine existed. The genes behind ABO did not appear with hospitals, laboratories, or microscopes. They were already moving through human bodies when our ancestors lived under shifting skies, when ice expanded and retreated, when grasslands opened and forest closed, when small groups survived by watching animals, seasons, and one another with unforgiving attention. Blood type then is not a neat historical document. It is more like a damaged manuscript written by evolution. Some pages are clear, others are torn. Scholars still debate exactly how much of today's blood type distribution reflects disease, how much reflects migration, how much reflects chance, and how much reflects ancient population bottlenecks, where a few survivors became the ancestors of many. No single explanation fully accounts for this pattern. That is what makes it so compelling. The story of blood type does not lead to one dramatic answer. It opens into a web of causes, each one tied to survival in a different way. Imagine a small prehistoric group moving along the edge of a vanished river. Their world is not primitive in the lazy sense. It is complex, alert, and dangerous. They know which roots can be eaten, which animals must be avoided, where stone can be found, when the dry season bites hardest. They do not know that their red blood cells carry inherited markers. They do not know that a fever spreading through their camp may spare some bodies more easily than others. They do not know that a trait passed quietly from one child to the next may become more common if it offers even a slight advantage against a disease that returns again and again. Evolution does not need intention. It only needs difference, pressure, and time. Across enough generations, even small differences can leave a mark. A blood type that slightly reduces the severity of one infection might become more common where that infection is relentless. Another type might offer no advantage there but persist because of chance isolation or movement from another region. A dangerous mismatch during pregnancy might affect family lines before anyone understands why some newborns sicken after birth. A population that passes through a bottleneck may carry forward whatever blood types happen to be present among the few who survived. The evidence remains incomplete, but the principle is clear. Human blood has been shaped by the same forces that shaped bones, skin digestion, immunity, and the ability to live in wildly different environments. This is where blood type becomes a doorway into the deeper human past. It points toward migrations out of Africa, toward ancient encounters among separated groups, toward farming villages where disease found new opportunities, toward cities where human density became both triumph and trap. It points toward the strange fact that civilization, for all its power, created new biological challenges. Settled life brought stored grain animals, waste crowding, and epidemic disease. Human bodies had to endure not only predators and hunger, but the invisible armies that followed people into their own success. And yet blood type cannot be treated like a secret code that explains everything. It does not reveal character. It does not determine intelligence, courage, love, or fate. It does not divide humanity into ancient tribes with pure and unchanging boundaries. Human history is far too tangled for that. Populations move, mix, disappear, return, absorb others, and are absorbed in turn. The same blood type can appear in many places for different reasons. The same ancestry can contain many blood types. The map is real, but it is not simple. Still, the map exists. Type O dominates in some parts of the world. Type B appears in striking patterns across Eurasia and parts of Asia. Type A has its own deep and uneven distribution. A B remains rare, a late combination in many populations. Rh-negative blood, especially common in parts of Europe compared with much of the world, has attracted both scientific interest and a cloud of unsupported speculation. Beneath the myths, there is a real evolutionary question. Why do these markers vary so sharply, and what old pressures helped arrange them? To follow that question, we have to move backward beyond modern blood banks and hospital charts, beyond national borders, beyond the idea that humanity has always looked the way it does now. We have to enter the time before blood had names, when these markers already existed in bodies that would never know them, and when evolution was quietly testing human survival in landscapes that have since vanished. Before blood has names, before it is sorted in glass tubes or labelled on hospital forms, it exists as an ancient biological surface crowded with signals too small for any prehistoric eye to see. These signals are not decorations. They are parts of the cell itself, built from molecules that sit on the outer membrane of red blood cells and other tissues exposed to the body's internal world and sometimes to the organisms trying to invade it. Long before humans understand inheritance, long before they speak of antigens or antibodies, these markers are already there, moving from one generation into the next, with the quiet persistence of a family story no one knows they are telling. The deeper origin of blood groups reaches beyond modern humans. It reaches beyond cities farming ice age shelters and painted caves. Some blood group variations are shared with other primates, which means their roots may be older than humanity itself. The ABO system in particular is not merely a recent human invention produced after people spread across the world. Related forms exist in other apes, suggesting that the basic genetic machinery behind these blood markers was already ancient before Homo sapiens appeared. This does not mean that a chimpanzee's blood type history is the same as a human one, or that every modern human pattern can be read backward into a simple primate past. The evidence remains incomplete, and the evolutionary paths have twisted in separate directions. But it does mean that blood type begins in a world older than our species, in bodies climbing, foraging, bleeding, breeding, and surviving under pressures that were already immense. To understand why such markers persist, it helps to picture the surface of a cell not as a blank wall but as a shoreline. Anything moving through the body must encounter it. Pathogens, immune molecules, and neighbouring cells all meet these surfaces. Some microbes use surface structures as handles, places to attach before entering or damaging tissue. If a tiny variation changes that attachment even slightly, it may influence who becomes gravely ill and who survives long enough to reproduce. Evolution often works through these small margins. It does not need perfection. It only needs a difference that matters often enough. Yet blood groups also survive through chance. Not every marker must have a dramatic purpose. Some may persist because they do little harm. Others may rise or fall because a small population expands after carrying certain variants by accident. Scholars still debate how much of the ABO story is driven by natural selection, and how much by neutral drift, the random reshuffling of genes through birth, death, isolation, and migration. The answer is almost certainly not one or the other. Human blood type history is a mixture of pressure and accident survival, and contingency adaptation and luck. In the long ages before our species, ancestral primates live in environments filled with parasites, wounds, seasonal shortages, and competition. A broken branch, a bite, a contaminated fruit, a fever from an insect-borne disease, each can become part of the pressure shaping bodies over time. Blood markers do not act alone in this world. They are only one part of immune defense, one piece of a much larger biological system. But because they sit at important contact points between the body and its threats, they become visible to evolution even when they remain invisible to the animals carrying them. When early members of the human lineage begin to walk through African landscapes, their bodies already contain old primate inheritances. The shape of the pelvis changes. Hands become more available for carrying. Teeth and jaws shift with diet. Brains grow through a long and uneven process. Stone tools appear. Fire eventually becomes part of daily life. Social bonds deepen, and survival depends more and more on cooperation. But inside the blood, older patterns continue. The body is never rebuilt from nothing. Evolution works by modifying what already exists. This is one of the most important truths in the story of blood type. Human evolution is not a clean upward ladder. It is a patchwork. Ancient features remain beside newer ones. Some are useful, some are harmless, some become risky under new conditions. A blood marker that once mattered in one ecological setting may later become neutral in another. A trait that offers a slight advantage against one disease may bring vulnerability to another. The same inheritance can be both shield and weakness depending on the world around it. Modern blood typing gives these inheritances tidy names, but the ancient reality is untidy. The ABO gene influences the presence of particular sugar molecules on cell surfaces. Different versions of the gene produce different patterns. Type A cells carry A markers. Type B cells carry B markers. Type AB carries both. Type O lacks functional A or B markers on red blood cells because of a mutation affecting the enzyme that would otherwise build them. That explanation sounds simple when placed in a modern chart, but its history is not simple at all. Each version has travelled through deep time, survived in bodies, exposed to changing landscapes, and entered different populations through paths that are only partly recoverable. What survives in the record suggests that some ABO variation is astonishingly old. This matters because it warns against a common mistake imagining that one blood type is the original pure or superior form of humanity. Evolution does not work that way. Old does not mean better. Common does not mean stronger. Rare does not mean mysterious in a supernatural sense. Each blood type is part of a long biological experiment without a designer shaped by many environments and carried by people whose lives were never reducible to the markers in their veins. There is also a quieter surprise. Blood group antigens are not found only on red blood cells. Many appear on other tissues and in bodily secretions depending on a person's genes. This means their evolutionary importance may involve more than transfusion, which is a modern medical issue. In deep history, no one is receiving stored blood from a stranger. The real ancient stakes are likely tied to infection reproduction and immune recognition. A molecule that seems important today because of blood transfusion may originally have mattered, because bacteria, viruses, or parasites interacted with it in the gut lungs, blood vessels, or reproductive system. The world before named blood is therefore not a blank prelude. It is already full of hidden biological conflict. Microbes evolve, hosts evolve. A pathogen that becomes better at exploiting one cellular marker may create an advantage for individuals with a different marker. Then over long periods the pathogen changes again. This back and forth can preserve diversity rather than eliminate it. No single blood type wins forever because the enemies are also moving. No single explanation fully accounts for this, but many scientists suspect that balancing pressures from disease helped keep multiple ABO types alive across immense spans of time. By the time Homo sapiens emerges in Africa, the blood story is already ancient, already complicated, already layered with inheritance older than the species itself. These early humans do not carry national identities, written histories, or fixed borders. They carry tools, children's scars, memories of waterholes and animal paths, and within their cells molecular signs inherited from ancestors stretching backward through vanished branches of the primate tree. Some of those signs will pass through bottlenecks, some will cross continents, some will become common in places their first carriers never imagined. The names will come much later, the maps will come later still, but the differences are already real. In the bodies of people walking beneath African skies, blood has begun a journey that will outlast climates, languages, kingdoms, and gods. It is not yet a medical category. It is an old surface facing an older world. The ancient ABO puzzle begins with something almost absurdly small, a change in a gene that alters the surface of a cell. No monument marks the moment. No fossil bone can show it clearly. Somewhere in the vast unrecorded past, inherited instructions shift and the red blood cells of one lineage begin carrying a slightly different molecular pattern from another. The body does not announce this difference. A hunter still runs. A mother still carries a child. A group still follows rain animals and ripening plants across a landscape that has no patience for weakness. Yet inside these bodies a quiet distinction has appeared, and over thousands of generations it will become one of the best-known human biological labels in the modern world. EBO blood type depends on inherited versions of a gene that helps build particular sugar structures on cell surfaces. Type A and Type B are not different colours of blood nor different qualities of life. They are different molecular endings, small chemical signatures attached to a broader structure. Type O, despite its later fame as the universal donor in many emergency transfusion contexts, is not a separate magical substance. It usually results from a version of the ABO gene that does not build a working A or B marker on red blood cells. Type AB appears when a person inherits one A version and one B version, so both markers are present. In the modern laboratory, this is clear enough. In the long prehistoric story, it becomes much stranger because these types are not inventions of one place or one people. They are old variations moving through shifting human worlds. For a long time, people imagined blood as a single human fluid with spiritual, symbolic, or family meaning. They were not entirely wrong to sense that blood carried inheritance, but they could not see the mechanism. Kinship, descent, and belonging were often described through blood, even though the actual markers that define blood groups remained hidden. The scientific discovery of ABO in the early 20th century did not create these differences. It merely exposed them. Suddenly the invisible had consequences that could be measured. Some blood mixed safely, some clumped dangerously. A transfusion could save a life only if the hidden markers were respected. Modern medicine discovered a rule that evolution had been writing for far longer than human memory. What makes ABO evolution difficult is that its main forms are ancient and widespread yet unevenly distributed. If one blood type were always superior, natural selection might have pushed it toward dominance everywhere. That did not happen. If the types were entirely neutral, their patterns might still vary, but certain signs suggest disease and environment may have influenced them. The evidence remains incomplete, and scholars still debate the balance between selection, chance, and migration. But the survival of multiple ABO types across deep time hints that each may have carried advantages, disadvantages, or harmless persistence under different conditions. Type A and Type B are produced by enzymes that modify cell surface molecules in different ways. These differences are tiny at the scale of a human life, but evolution often turns on tiny differences repeated across enormous time. The O version, especially common in many populations today, is often linked to a mutation that disrupts the enzyme's function. That means the cell surface does not display the A or B marker in the usual way. In one setting, that absence might be helpful. In another, it might not matter. In another still, it might create vulnerability. The strange power of ABO is that it does not tell a single story. It tells several at once. Consider the earliest Homo sapiens populations in Africa. They are not a single tribe moving as one body. They are scattered, diverse, and changing over time. Some groups live near coasts and rivers. Others move through savannas, woodlands, uplands, and dry corridors that expand or shrink with climate. Populations separate and reconnect. Some lineages vanish, others merge. Ancient genetic variation is constantly being filtered through survival and accident. In such a world, a blood type can rise in one group not because it is universally better, but because that group happens to grow, migrate, or survive a crisis while carrying it. Population bottlenecks matter here. When a population is reduced to a smaller number of surviving individuals, the genetic future can be shaped by what those survivors happen to carry. A rare variant may become common. A common one may disappear from a region. Later generations may look at the pattern and search for a grand explanation, when part of the answer may be a narrow passage through hunger, drought, disease, or isolation. No single explanation fully accounts for this. But bottlenecks are one reason human genetic maps often look like the result of both history and accident. Still, chance cannot explain everything. Infectious disease is one of the strongest suspects in the ABO story because many pathogens interact with cell surfaces. Some research suggests associations between ABO type and vulnerability or resistance to certain infections. These associations are rarely simple, and they should not be overstated. A blood type does not make a person invincible, nor doomed. But over many generations, even a small difference in survival during repeated outbreaks can shift the frequency of a blood type in a population. Evolution is not impressed by individual certainty. It works through probabilities. The ancient world is full of such probabilities. A child survives a fever that kills another. A pregnant woman lives through an infection that others do not. A village endures a wave of disease with slightly fewer losses because some inherited trait reduces severity. No one understands why. No one names the trait. But the survivors have children, and their children carry the same hidden markers. Over centuries, the pattern thickens. Over millennia, it becomes part of a regional signature. The ABO puzzle is also complicated by the fact that humans do not stay still. Groups expand out of Africa, encounter new climates, mix with other human populations, cross coastlines, follow herds, settle valleys, and later build farms, towns, and empires. Every movement rearranges blood type frequencies. Every isolation preserves certain patterns. Every mixing blurs them. The result is not a clean diagram, but a palimpsest one layer written over another. A modern distribution map may show high frequencies of one type in a region, but that pattern may reflect many events, ancient founder effects, later migrations, disease pressures, conquest trade, or simple demographic growth. ABO also resists the desire for hierarchy. Some popular accounts try to attach blood types to ancient lifestyles, claiming one belongs to hunters, another to farmers, another to nomads. These stories are often too neat. They may contain a faint echo of real population history, but they usually stretch far beyond the evidence. What survives in the record suggests a far more tangled reality. Hunters could carry any ABO type. Farmers could carry any ABO type. Nomads, sailors, kings, slaves, monks, and merchants all carried mixtures shaped by ancestry and chance. Blood type can illuminate human evolution, but only when it is treated as a clue, not a verdict. The deeper truth is more interesting than the myth. ABO types are old because diversity itself may have been useful. A population with varied blood markers may be harder for a single pathogen to exploit completely. If a disease is especially effective against one marker, individuals with another may have a better chance. Over time, this can help preserve multiple forms in the species. The evidence remains debated, but the idea fits a broader pattern. In immune-related evolution, diversity can be a defense. A uniform population may be efficient in one environment, but fragile when the enemy changes. So the ABO puzzle is not simply why humans have different blood types, it is why those differences have endured. Through ice ages, migrations, extinctions of neighbouring human species, the rise of agriculture, the birth of cities, and the spread of epidemic disease, ABO and later AB remain part of the human inheritance. They move silently through royal bloodlines and forgotten graves, through isolated villages and crowded ports, through people who leave monuments and people whose names disappear entirely. Each type is both ordinary and ancient, carried by millions, yet rooted in a past almost too deep to imagine. By the time human groups begin spreading widely across the planet, the ABO system is already waiting inside them. It will not determine their cultures, their languages, or their destinies, but it will travel with them, and as they cross deserts, mountains, rivers, ice, and sea, Geography will begin to sort these invisible markers into patterns that still haunt the modern map. As human beings move outward across the planet, they do not travel as empty figures on a map. They carry fire memory tools, fear, hunger, children, parasites, stories and genes. They carry the marks of ancestors whose names are gone beyond recovery. Among those marks are blood group variants invisible to the travellers themselves, yet faithfully moving with them, from valley to coast, from forest to steppe, from river mouth to island shore. In this movement, geography begins to do what it has always done to life. It separates, filters, traps, protects and reshapes. Blood types become part of that wider sorting, not because mountains or oceans choose them, but because human groups are small, vulnerable and often isolated enough for chance and survival to leave lasting signatures. The first great stage of this story unfolds in Africa, where Homo sapiens emerges within a continent of enormous ecological variety. Africa is not a single backdrop. It contains wetlands, dry grasslands, tropical forests, highlands, coasts, lakes, and deserts that expand and shrink as climate changes. Early human populations do not simply leave one birthplace in a single heroic march. The evidence remains incomplete, but what survives in genetics and archaeology suggests a long, complex African prehistory, with groups dividing, reconnecting, and carrying different pieces of human variation through time. Some blood group diversity may have been shaped in this deep African setting before any major movement beyond the continent. When some human groups eventually expand into Eurasia, they pass through narrow corridors of possibility. Climate opens and closes routes. Sea levels rise and fall. Deserts become barriers, then grasslands, then barriers again. A population that leaves one region may represent only a fraction of the diversity left behind. This is the founder effect when a small group gives rise to a larger population. The genetic traits common among the founders can become common among their descendants, even if those traits were not especially advantageous. In blood type history, founder effects matter because they can make an ordinary accident look thousands of years later like destiny. A few families carrying certain ABO frequencies enter a new region. They survive. Their descendants multiply. Other groups disappear, merge, or are absorbed. Over time, the blood type pattern of that region may partly reflect the blood of the founding group, rather than some perfect adaptation to the landscape. Yet the landscape still matters. It determines who can cross, who remains cut off, which diseases flourish, what foods are available, how many people can live there, and how often outsiders arrive. Geography does not write the whole story, but it holds the page steady while history leaves its marks. Ice Age Eurasia is especially important because it is not the familiar world of modern maps. Vast ice sheets cover northern regions. Sea levels drop, exposing lands now drowned. Deserts shift. Mammoth steppe stretches across immense spaces, cold and dry, supporting animals that no longer exist. Human groups live in scattered pockets, sometimes separated by climate barriers for long periods. In such conditions, genetic drift can be powerful. A blood type present in one small group may become common simply because that group survives a harsh interval while another does not. No single explanation fully accounts for the patterns, but repeated isolation and expansion are essential parts of the story. Then there are islands and remote regions where blood type can become especially striking. When a small number of people settle an island, they bring only a sample of the genetic variation from their homeland. If the island remains isolated, that sample can become exaggerated. A blood type rare in the source population might become more visible. Another might be absent altogether. This does not require mystery in the supernatural sense. It requires boats, chance, distance, and time. Human island histories are full of such founder effects, though later contact often complicates the picture. The Americas provide one of the most dramatic examples of blood type distribution shaped by ancient migration and isolation. Many indigenous populations of the Americas have very high frequencies of type O, though the exact reasons remain debated. Some of this may reflect founder effects among the ancestral populations that entered the Americas from Northeast Asia. Some may involve later population bottlenecks, isolation disease pressures, and the catastrophic demographic collapse after European contact. The evidence remains complex and cannot be reduced to one cause, but the pattern shows how blood type can become a surviving trace of long journeys, narrow passages, and devastating historical events. In Eurasia, the picture is more mixed. Type B appears at higher frequencies in parts of Central Asia, South Asia, and Northern Asia, while being less common in many Western European populations. Type A is frequent in many European regions and appears elsewhere in varied patterns. Type O remains widespread across much of the world. These distributions do not map neatly onto language, culture, or ancient identity. They are older than many nations and younger than some migrations layered by repeated movements. Steppe expansions, farming dispersals, trade routes, invasions, and local population histories all contribute to the present pattern. The spread of farming adds another turning point. As agriculture expands from several independent centres, human populations grow denser. Villages become towns. Stored food supports larger communities, but it also attracts rodents, insects, and contamination. Domesticated animals live close to people. Waste accumulates. Water sources are shared by many. Disease ecology changes profoundly. Blood types moving through farming populations are no longer facing only the pressures of small mobile bands. They're now travelling through crowded human landscapes where infection can spread in new ways. What was once a marker shaped by older environments now enters the dangerous experiment of civilization, yet migration continues to blur every boundary. Farmers move into lands occupied by hunter-gatherers. Sometimes they replace local populations, sometimes they mix with them. Pastoralists expand across open grasslands. Seafarers stitch islands into networks. Merchants cross deserts. Armies move faster than families, but families follow. Captives are taken. Empires rise and collapse. Every movement carries blood markers into new combinations. A region's blood type pattern can preserve ancient traces, but it can also be transformed repeatedly. The map is not a fossil frozen in one age. It is a living surface scraped and rewritten. There is a temptation to look at blood type maps and draw hard borders around peoples. That temptation should be resisted. Blood type frequencies describe populations statistically. They do not define individuals or cultures. A person with type B in Europe is not a living remnant of one simple migration. A person with type O in the Americas is not explained by one prehistoric event alone. A person with type A in Asia or Africa does not carry a single story that can be read without context. Blood groups can suggest pathways, but they cannot replace archaeology, language, history, ancient DNA, or the lived complexity of human communities. Still, when handled carefully, these patterns are powerful. They remind us that human evolution did not stop when Homo sapiens appeared. It continued as people moved into new ecologies and built new ways of life. Blood type frequencies are one of many clues showing that our species has never been biologically uniform. We are connected by common ancestry, but we're also shaped by local histories of isolation, exposure, survival, and mixing. The same bloodstream that makes humanity one also records the long unevenness of the human journey. By the time villages thicken into towns and towns into cities, the old markers have already travelled far from their earlier landscapes. They have crossed Ice Age plains, tropical coasts, dry basins, mountain passes and sea routes. They have been sorted by geography and shuffled by movement. But a new force is rising beside geography, one even more relentless than distance. It is disease, and in the crowded human world, disease will begin pressing against blood type, with a force that may help explain why some markers survive spread or remain strangely rare. Disease is the oldest empire humanity has ever lived under. It has no capital, no throne, no army visible on the horizon, yet it follows people wherever they gather, drink, touch, trade, bury, butcher, farm, and flee. In the deep past, long before anyone can see bacteria, viruses, or parasites, human beings still know their power. A settlement that seems strong in the dry season can become a place of mourning when fever arrives. A river that gives life can carry death downstream. A caravan can bring salt copper stories and infection in the same breath. And inside these outbreaks, inside the repeated pressures that strike generation after generation, blood type may have become one of the quiet variables in who survives. The link between blood groups and disease is not simple. It is not a clean rule in which one type is protected and another is condemned. Modern studies often reveal associations, not absolute destinies. A blood type may slightly raise risk for one illness, slightly lower risk for another, and make no meaningful difference for many more. The evidence remains incomplete, and scholars still debate how much disease shaped the global pattern of ABO types. But the possibility is difficult to ignore because pathogens often interact with the surfaces of human cells, and ABO markers are part of that surface world. The same tiny molecular flags that matter in transfusion can also matter when an invader searches for a foothold. Cholera offers one of the clearest and most unsettling clues. In modern research, people with type O have often shown greater vulnerability to severe cholera symptoms compared with some other blood types. Cholera is a disease of water crowding contamination and speed. It can empty the body with terrifying force. In ancient and historical communities where clean water systems are absent or fragile, repeated outbreaks could have placed intense pressure on populations. If type O increased the risk of severe disease in cholera-prone regions, then over long periods it might have been selected against there. This does not prove a single global pattern, and cholera's deeper history is difficult to reconstruct perfectly, but it shows how blood type could matter in a world where water itself could turn hostile. Malaria presents another layer, though its relationship with ABO is more complicated. Some research suggests that type O may offer a degree of protection against severe forms of malaria, particularly by reducing certain mechanisms that allow infected red blood cells to clump and obstruct blood flow. This possible advantage could help explain why type O remains common in some regions where malaria has been a heavy historical burden. Yet malaria is not one disease in one place. It includes different parasites, ecological settings, mosquito species, and human genetic defenses. Sickle cell trait thalassemias, G6PD deficiency, Duffy negativity, and other adaptations also shape the malaria story. ABO is only one thread in a much larger net. No single explanation fully accounts for survival under malaria, but the disease shows how violently natural selection can act when infection returns century after century. Smallpox, plague, tuberculosis, leprosy, influenza, and other infections belong to the same broad landscape of pressure, though the exact connections to blood type are harder to prove. Some associations have been proposed, some remain uncertain, and others may weaken when studied more carefully. This is where the story must stay grounded. It is tempting to imagine a hidden blood type code behind every epidemic, but human disease is never that tidy. nutrition age, previous exposure, social conditions, sanitation housing, climate immunity and genetics all interact. Blood type may tilt the odds, but it rarely writes the whole outcome. Still, even a tilted odds table can become powerful in deep time. A difference too small to feel in one generation can become visible after 1,000. If a blood type reduces fatality by a few percent during repeated outbreaks, more carriers may survive to have children. If another type slightly increases vulnerability in a region where a disease is constant, it may become less common. Evolution does not require certainty. It works through the accumulation of small advantages and disadvantages under relentless repetition. In this sense, disease may have been one of the great invisible sculptors of human blood. The rise of agriculture intensifies this pressure. Small mobile bands can suffer from parasites and infections, but many crowd diseases struggle to persist when human groups are too small and dispersed. Farming changes that. People live closer together, they store grain, they share wells, they keep animals nearby. Waste builds around settlements. Rodents and insects find new habitats. The human body enters a more crowded microbial world. What civilization offers in food surplus and social complexity, it often takes back in fever. The triumph of settlement is also the creation of new disease corridors. Imagine an early farming village after rain. Mud thickens between houses. Smoke hangs low under thatched roofs. Animals breathe near sleeping spaces. Children run barefoot through ground contaminated by waste. Grain stores attract mice. Water jars are refilled from a source used by many families. No one knows why illness spreads, but everyone knows when it has arrived. In such a place, inherited differences that affect infection may become meaningful. Blood type alone cannot determine survival, but it becomes part of the biological background against which disease acts. Trade makes the situation larger. Once villages connect to towns and towns connect to routes, pathogens gain roads. A sickness that once might burn out locally can travel with merchants, soldiers, pilgrims, sailors, and captives. Ports become gateways not only for goods, but for microbes. Empires with their roads and armies create vast networks through which disease can move. The same systems that spread language, coinage, law, and religion also spread infection. Human success gives disease a larger stage. There is a grim irony in this. The human traits that make civilization possible, cooperation, density, exchange, memory, and mobility, also make epidemics more dangerous. Blood types inherited from deep evolutionary time are carried into this new world and tested under conditions their earliest carriers never faced. A marker that once interacted with local parasites may now encounter diseases from distant regions. A population once isolated may suddenly face pathogens to which it has little inherited resistance. The old blood map is disturbed by the new disease map. The catastrophe after European contact in the Americas shows how devastating disease can be when populations meet after long separation. Smallpox and other introduced infections killed on a scale that reshaped continents. Blood type alone does not explain this tragedy. Lack of prior exposure, social collapse, violence, forced labor, famine, and colonial disruption were central. But the event reminds us how much human history has been governed by immune experience. Populations carry not only genes but histories of exposure. When those histories collide, the results can be catastrophic. In older periods, similar but smaller collisions likely happened many times. Expanding farmers met foragers. Steppe pastoralists entered settled lands. Armies returned from distant campaigns. Ships arrived from infected ports. Each contact rearranged the microbial world. Some people survive because of prior immunity. Some survive because of health, luck, age, or care. Some may have survived because inherited traits, including blood group markers, gave them a slight advantage. What survives in the record suggests that no ancient community was biologically untouched by disease pressure. The immune system itself is a history of conflict. It learns, remembers attacks, overreacts, and sometimes fails. Blood group markers sit at one edge of this larger system, not as commanders, but as features in the terrain. A pathogen meets the body. As an invading force meets a landscape, some surfaces offer paths, some barriers, some traps, some opportunities. Change the landscape slightly, and the invasion may unfold differently. Over time, those slight differences can shape populations. This is why blood type is so revealing when viewed through evolution. It does not show a heroic story of perfect adaptation. It shows compromise. Type O may help in one disease context and hurt in another. Type A may carry one risk and another possible advantage. Type B may follow its own uneven trail. A B may remain rare, partly because it requires the meeting of A and B lineages, but its health associations are still complex. There is no champion blood type standing above the rest. There are only different inheritances surviving different worlds. By the time the ancient disease landscape has done its work, the blood map is already marked by contradiction. Type O can be extremely common, yet not universally advantageous. Type A can flourish in dense regions, yet face its own risks. Type B can spread widely across some parts of Eurasia, while remaining uncommon elsewhere. The forces behind these patterns are not always visible, but disease leaves fingerprints where no bones survive. It presses on the living, and the living pass forward whatever allowed them to endure. The long shadow of disease does not solve the mystery of blood type, but it deepens it. Blood is not simply a family inheritance. It is a battlefield record written in molecules rather than scars. Every modern blood group frequency may carry some echo of ancient fevers, contaminated waters, insect bites, crowded villages, and desperate survivals no chronicle ever preserved. And among all the blood types shaped by this invisible struggle, one stands out for its reach, its medical importance, and its strange double nature. Type O, common across much of the world, powerful in transfusion, possibly protective in some settings, and dangerously vulnerable in others. Type O has a way of attracting simple stories. It is common, medically famous, and often described in language that makes it sound almost primal, as if it were the original human blood standing untouched beneath all later variation. But the real story is more elusive. Type O is not a badge of purity, not a sign of superiority, and not a biological crown passed down from some perfect ancestral people. It is a version of the ABO system shaped by mutation, inheritance, drift disease, and migration, and its power lies partly in contradiction. In one setting it may be useful, in another it may be costly. Across the long history of humanity, Type O survives, not because it always wins, but because evolution rarely works by producing anything so clean. At the surface of the red blood cell, Type O is defined by absence. The basic structure is there, but the A and B markers are not added in the usual functional way. That absence becomes enormously important in modern transfusion medicine because Type O red blood cells lack A and B antigens that would trigger dangerous reactions in many recipients. This is why Type O negative blood, when Rh is also considered, becomes so valuable in emergencies. Yet this modern usefulness can mislead the imagination. Prehistoric people were not doing transfusions beside Ice Age fires. The evolutionary success of Type O must have come from older forces, survival, reproduction, migration, and chance. One of the most striking things about Type O is its high frequency in many populations across the world, especially in parts of the Americas, Western Europe, and Africa. But high frequency does not mean one cause. In the Americas, type O's dominance among many indigenous populations likely reflects founder effects bottlenecks and long isolation, later complicated by the terrible population losses after European contact. In parts of Africa, type O may intersect with disease pressures such as malaria. In Europe, its frequency reflects layers of ancient and later population history. The evidence remains incomplete, and scholars still debate the relative weight of these causes. Type O is common, but commonness is not an explanation by itself. Its possible relationship with malaria gives the story a darker edge. Severe malaria can turn red blood cells into instruments of their own destruction. In some forms of the disease-infected cells stick to other cells and to blood vessel walls, creating dangerous blockages that can damage organs and kill. Some studies suggest that type O red blood cells may be less prone to certain forms of clumping associated with severe malaria. If that protection is real and significant across generations, it could help type O persist or increase in regions where malaria has been a relentless killer. But malaria has many genetic countermeasures, and type O is only one possible factor among many. No single explanation fully accounts for the blood map of malarial regions. Then comes the reversal. Type O may be associated with worse outcomes in severe cholera, a disease that strips water from the body with brutal speed. In places where cholera or cholera-like diarrheal diseases repeatedly strike, this could work against type O. Here, the same blood type that may offer one kind of protection becomes a liability under another microbial regime. That contradiction is not a flaw in the theory. It is the point. Evolution does not choose traits for a universal world. It tests them against local dangers, and the dangers change. This double nature helps explain why type O can be both widespread and uneven. A trait may spread in one ecological context or in another, and survive by chance in a third. A population may carry high levels of type O because its founders did not, because type O offered any special local advantage. Another population may lose some O frequency, because disease pressures made other types slightly more favourable. Later migration can scramble the pattern again. What survives in the record suggests that type O is not the answer to human evolution, but one of its most persistent clues. There is also a quiet reproductive dimension. A bioincompatibility between mother and fetus can sometimes influence pregnancy, though its effects are usually less dramatic than Rh incompatibility. In some cases, a mother's immune system may react to fetal blood group markers inherited from the father. This can contribute to newborn jaundice or other complications, though modern medicine usually manages these risks. In deep history without diagnosis or treatment, even modest reproductive pressures might have mattered over long periods. Still, the evidence linking ABO incompatibility to large evolutionary shifts remains uncertain. It is one more piece of a complicated puzzle, not a master key. The fame of type O has also fed popular myths. Some claim it is the blood of ancient hunters, the blood of strength, the blood of a more original humanity. These ideas are too neat and often unsupported. Hunter-gatherer populations carried varied genetic histories, and modern blood type does not preserve a person's ancient lifestyle like a label pinned to the body. A farmer could be O, a hunter could be A herder could be B, a city dweller could carry any type at all. Blood type is inherited through family lines, not through moral character or romanticized ancestry. It can reveal population patterns, but it cannot turn individuals into living museum specimens. The more grounded version is far more interesting. Type O is a story of absence becoming meaningful. A missing molecular marker changes how the immune system recognizes blood, how transfusion works, and perhaps how some pathogens interact with the body. That is the sort of strange economy evolution often uses. A loss can become a strength. A weakness can persist because it is tied to another advantage. A mutation that looks like a broken instruction may become common because the world rewards it under certain conditions. In ancient communities, no one can see any of this. A child with type O is not marked by appearance. A woman with type O does not know that her red blood cells lack A and B markers. A dying man in a fever hut does not know whether his blood type has altered his odds by a small margin. The marker remains invisible, but invisibility does not mean insignificance. Generations pass and the survivors carry forward whatever combination of traits luck, care, and circumstance allowed them to remain. Type O's modern medical role gives the ancient story a final irony. A blood type shaped by forces no one understood becomes in the age of hospitals one of the most urgently needed gifts one person can give another. The same absence of A and B markers that may have mattered in old disease landscapes now allows type O red cells to cross boundaries between bodies in moments of trauma. A prehistoric molecular accident becomes a modern instrument of rescue. But it is important not to confuse medical usefulness with evolutionary destiny. The blood bank did not make type O common. It revealed one consequence of its biology. By following type O, the human story becomes less like a straight line and more like a landscape of trade-offs. What protects in marshland may harm in river cities. What spreads through a founder population may remain common long after the original circumstances vanish. What seems universal in one region may be rare in another for reasons buried under migrations and extinctions. Type O endures because human history itself is uneven, and because survival is rarely about perfection. It is about enough. So type O stands at the center of the blood type mystery, not as the oldest ruler of all blood, but as a witness to the strange compromises of evolution. It carries hints of malaria, echoes of bottlenecks, traces of migration, and the modern burden of emergency medicine. Its story is powerful because it refuses to be simple, and once that is understood, another blood type comes into view with a very different pattern, less globally dominant, more unevenly distributed, and closely tied in the popular imagination to the great corridors of Eurasia, where steppe, mountain, desert, and empire repeatedly rearrange the human map. Type E enters the story like a trail crossing a continent, visible in places, faint in others, widening across some regions of Eurasia, and narrowing as it approaches others, never forming a perfect line, never obeying the clean borders that maps later impose. It is less common globally than type O, and less evenly spread than type A. But in certain populations it appears with striking frequency. That unevenness has made it a favorite subject for speculation, especially because its distribution seems to brush against the long routes of herders, traders, conquerors, and migrants who move through Asia's interior. Yet the real history of type B is not a single march from one homeland. It is a layered pattern of ancient variation, population movement, disease pressure, drift, and repeated mixing across the largest landmass on Earth. At the molecular level, type B differs from type A by the specific sugar structure added to the cell surface. The difference is tiny, but it is enough for the immune system to recognize one marker as distinct from the other. A person with type B can safely carry B markers, but will usually have antibodies against A markers. This basic medical fact is clear in modern blood typing. The evolutionary meaning is less clear. Scholars still debate why type B reaches relatively high frequencies in parts of Central Asia, South Asia, and parts of Northern and Eastern Asia, while remaining less common in many indigenous American populations and lower in much of Western Europe. No single explanation fully accounts for this uneven map. One part of the answer may lie in old population structure. Eurasia has never been a quiet landmass. During the Ice Age and after groups repeatedly expanded, retreated, isolated, and reconnected. Mountains divided populations. Grasslands opened corridors. Deserts acted as barriers in some centuries and bridges in others. The steppe especially became a vast human highway once pastoralism developed. People with herds could move across distances that would have been punishing for settled farmers. As these groups travelled, mixed raided, traded and settled, they carried blood types with them. Type B may have moved through some of these networks not as the mark of one people, but as one inherited variant repeatedly redistributed by movement. The popular imagination often links Type B to nomads of the Eurasian steppe. There may be a faint historical reason for the association, because some populations connected to Central and Inner Asia have had relatively high frequencies of Type B. But the simplified version quickly becomes misleading. The steppe was not one tribe. It was a shifting world of languages, confederations, marriages, captives, alliances and replacements. A blood type cannot identify a person as a nomad, and it cannot prove descent from any particular empire. The evidence remains incomplete, and modern distributions reflect many episodes, not one. Type B's trail across Eurasia is less like a military road, and more like braided river channels splitting and rejoining over thousands of years. South Asia adds another layer. Type B is relatively common in many South Asian populations where human genetic history is deeply complex. Ancient hunter-gatherer ancestry, Iranian-related agricultural ancestry, steppe-related ancestry, local founder effects caste, and community endogamy and regional disease environments have all contributed to present-day genetic structure. Blood type frequencies are part of that complexity, but they cannot untangle it alone. What survives in the record suggests that Type B's presence there may reflect both ancient and later demographic layers shaped by population mixture and social boundaries that sometimes preserve differences for long periods. In East Asia, Type B appears in varied frequencies, again resisting any simple explanation. Movements of farmers, pastoralists, northern groups, southern groups and later historical populations all contributed to the pattern. The rise of rice farming, millet farming, bronze technologies, state, systems, trade routes and frontier interactions created repeated opportunities for genes to move. Type B may have travelled with some of these expansions, but it also may have been present in older populations. Ancient DNA is beginning to clarify pieces of this picture, yet many gaps remain. The living map is not enough by itself. It must be compared with remains from the past. Disease may also have played a role, though here the story becomes uncertain. Some blood types appear to influence susceptibility or resistance to certain pathogens, but Type B's evolutionary advantages and disadvantages are not as popularly fixed as those claimed for Type O. Associations have been studied for various infections, yet many are context-dependent or not strong enough to explain global patterns alone. It is possible that in some regions Type B persisted because it carried modest advantages against local diseases. It is also possible that much of its distribution reflects demographic history more than selection. The honest answer is that scholars still debate the balance. This uncertainty is not a weakness in the story. It is part of what makes Type B revealing. It reminds us that not every surviving trait needs a dramatic survival legend. Some traits endure because they are good enough. Some rise because their carriers happen to move at the right time. Some become common in communities that remain socially or geographically isolated. Some spread through empire not because empire selects for them, but because empire moves people. Human evolution is not only a story of adaptation, it is also a story of contingency. Picture a chain of ancient movements across Eurasia. Families following herds beyond the horizon, farmers expanding along river valleys, traders crossing mountain passes with cloth and metal armies, dragging captives from one region into another, refugees leaving burned settlements, marriages arranged between groups that once spoke different tongues. None of these people thinks of Type B. They think of pasture-grain debt, honor, fear, hunger, and survival. Yet with every birth, the hidden marker either passes forward or disappears. Over time, human choices and human disasters become genetic patterns. Type B's unevenness also shows how difficult it is to separate ancient from historical time. A modern population may have a blood type frequency shaped partly by events 10,000 years ago and partly by events only 1,000 years ago. The spread of Turkic-speaking groups, Mongol expansions, Silk Road exchanges, Indo-European migrations, Islamic-era trade, local marriage systems and colonial movements can all leave traces that overlap older patterns. Blood type does not come with a timestamp. It must be interpreted cautiously beside other evidence. There is a lesser-known detail here that complicates the map even more. Blood group frequencies can vary sharply between neighbouring communities, especially when marriage has long occurred within social, religious, linguistic, or geographic boundaries. Two villages may share a region and climate yet differ because their ancestors married mostly within separate networks. In such cases, blood type reflects not only continental migration, but the intimate structure of community life. The grand sweep of Eurasia is written in part through household rules, local boundaries, and repeated choices of kinship. For the listener, seeking a mystery type B offers one, but not the kind that demands a lost race or hidden civilization. Its mystery is ordinary and deeper. How does a tiny cell surface marker become distributed across the world in a pattern that seems meaningful, yet refuses a single meaning? The answer is that human history itself refuses a single meaning. People move for climate, animals, soil, marriage, trade, fear, ambition, and force. Genes follow. Then disease, chance, and isolation edit the results. Type B therefore stands as a reminder that blood type is not a pure relic of prehistory alone. It is a living archive that includes deep ancestry and later turbulence. It passes through the great open spaces of Eurasia, through farming basins, mountain corridors, caravan cities, and imperial frontiers. It is carried by people who leave inscriptions and by people who leave nothing but descendants. Its pattern is not a sentence written once, but a manuscript revised by every age. As the blood map continues to unfold, another type draws attention for a different reason. Type A is not rare like AB, nor dominant everywhere like O, nor as geographically suggestive in the popular imagination as B. But its story becomes especially compelling when humans settle farm crowd together and create the dense worlds where civilization begins to change, not only culture, but the biological pressures surrounding every human body. Type A belongs to a different kind of landscape in the human imagination. It does not usually summon the vast steppe trails associated with Type B, or the sweeping global dominance often attached to Type O. Instead, it seems to settle into fields, villages, coasts, old farming zones, and dense populations where human life becomes more rooted, more crowded, and in many ways more dangerous. This does not mean Type A was born from agriculture, and it does not mean every person with Type A descends from farmers in any simple way. The deeper reality is older and more tangled. But Type A becomes especially interesting when viewed against the rise of settled worlds, because farming does not merely change what people eat. It changes the entire disease environment around the body. The A marker, like the B marker, is built by an enzyme that modifies the surface of red blood cells and other tissues. The difference is molecular, not visible. Yet that small surface structure becomes part of a much larger story when people begin living in dense communities. Farming populations grow because cultivated plants and domesticated animals can support more children per acre than hunting and gathering in many environments. More children survive weaning. More people live close together. Villages become persistent places rather than temporary camps. Houses stand beside houses. Paths become worn into the soil. Storage pits, animal pens, hearths, and waste areas remain in use for years. The human body enters a new ecological trap made by its own success. This is one of the great turning points in human evolution after the appearance of Homo sapiens. Evolution does not stop when culture begins. Culture changes the pressures that evolution must respond to. Agriculture creates new diets, new labor patterns, new social hierarchies, new forms of violence, and new diseases. The teeth of early farmers often show more cavities than those of many earlier foragers, because starch-heavy diets feed oral bacteria. Bones sometimes show signs of nutritional stress, repetitive labor, and infection. Settled life is not simply progress. It is an exchange, and the price is written into skeletons as well as blood. Type A has sometimes been linked in popular accounts to agricultural peoples, but this claim is too simple when stated as a rule. Type A existed before farming, and many non-farming populations carried it. Farming did not create the allele from nothing. Still, the expansion of farmers could have helped spread certain blood-type frequencies if the farming populations already carried them at notable levels. In Europe, for example, the spread of agriculture from Anatolia and the Near East contributed greatly to the ancestry of many later populations, though it later mixed with hunter-gatherer and steppe-related ancestry. If those farming groups had particular ABO frequencies, their demographic expansion could have influenced the later blood map. The evidence remains incomplete, but the mechanism is plausible, not because type A made people farmers, but because farmers carrying type A multiplied and moved. Dense settlements also create a possible arena for disease selection. Some studies have linked type A with different risks for certain infections and health conditions, though such associations are often complex and should not be inflated into destiny. In a world of early villages, however, even small differences might matter. A pathogen spreading through contaminated water or close contact could interact with cell surface markers in ways that slightly alter infection or severity. Over many generations, such pressures may nudge frequencies. Scholars still debate which pathogens mattered most and whether type A's regional patterns are better explained by migration selection or drift. The safest answer is that all three likely played roles in different places. The farming world is also a world of animals. Goats, sheep, cattle, pigs, chickens, dogs, and later horses live near people, each bringing its own microbial world. Many major human infectious diseases have connections to animals, though the pathways are varied and often complex. When people sleep near livestock, breathe their dust, handle their waste, drink their milk, butcher their bodies and share insects with them, microbes gain opportunities. Some fail to cross the species boundary, others adapt. Over time, settlements become laboratories of contact, not in the clean modern sense, but in the muddy, smoky, relentless sense of daily life. In this setting, blood type may be one part of the body's front line. The A marker appears not only on red blood cells but in many people on tissues and secretions, where microbes first encounter the human body. That means its ancient significance may have involved respiratory, digestive, or reproductive surfaces as much as blood itself. A pathogen that recognizes or mimics certain host molecules may behave differently depending on ABO type. The evidence remains incomplete, but the possibility shows why blood groups are more than transfusion categories. They are part of the body's interface with the world. There is a lesser-known complication in the ABO system. Some people are secretors, meaning their ABO antigens appear in bodily fluids such as saliva and mucus, while non-secretors do not express them in the same way. This secretor status depends on another gene, not the ABO gene itself, and it can influence interactions with certain pathogens. In deep history, such differences may have mattered in the gut, mouth, and airways, especially as diets and settlement patterns changed. Blood type, then, is not a single isolated marker. It belongs to a network of surface chemistry that shapes how bodies meet microbes. The growth of settled worlds also changes reproduction. Larger communities mean more possible partners, but also more social rules around marriage, inheritance, and status. Some groups become endogamous, marrying mostly within defined boundaries. Over time, this can preserve or exaggerate local blood type frequencies. A village caste, clan, island, valley, or religious minority may develop a distinctive pattern, not because the blood type itself caused the social boundary, but because the boundary shaped inheritance. The story of type A in settled populations may therefore include both biological and cultural filters. Civilization makes this even more intense. Once cities appear, disease no longer needs to wait for rare contact between small bands. It has streets, markets, temples, barracks, wells, canals, and ships. A city is a magnificent human achievement, but from the viewpoint of a pathogen, it is also a feast. Type A, Type B, Type O, and eventually AB all circulate through these urban bodies, exposed to waves of infection that would have been difficult to sustain in sparse prehistoric populations. The old blood markers are now tested in a new theatre built from mud brick, stone grain lore, and crowding. Yet Type A's story should not be mistaken for decline or doom. People with Type A survived, reproduced, built, migrated, ruled, farmed, sailed, wrote, fought, and endured like people with every other blood type. The associations between blood type and disease are not individual prophecies. They are population-level hints, often small and context-dependent. The danger of blood type storytelling is that it can turn statistical tendencies into personal myths. The better use is humbler to see how tiny inherited differences move through vast historical forces. Type A in this light becomes a marker of entanglement. It is ancient, yet its distribution may have been reshaped by farming expansions. It is biological, yet its frequencies can be influenced by cultural marriage rules. It may interact with disease, yet disease itself is shaped by housing, water, animals, climate, and trade. It sits at the crossing point where evolution and civilization meet. What survives in the record suggests that settled life did not simply shelter humanity from nature. It created new versions of nature inside the village wall. By the time early cities rise along rivers and coasts, blood types have already become part of a denser human experiment. Type O carries its paradoxes. Type B follows its uneven Eurasian trails. Type A moves through ancient and settled worlds with its own complicated legacy. But one ABO type remains different because it requires a meeting. It appears only when A and B lineages come together in the same family line, producing a blood type that is medically distinct, globally rare, and evolutionarily revealing precisely because it is born from mixture. A B is the rarest of the familiar ABO blood types, and its rarity gives it an atmosphere of mystery that is easy to exaggerate. It is sometimes described as modern, unusual hybrid, or strangely recent, as if it were a new arrival in human blood. There is a partial truth hidden in that impression, but it must be handled carefully. AB is not a separate ancient blood lineage in the same way people imagine AB or O. It appears when a person inherits an A allele from one parent and a B allele from the other. It is a meeting place, not a single path. Wherever A and B have long existed together in a population, AB can appear. Wherever one of them is rare or absent, AB remains rare as well. This makes AB a blood type of contact. It depends on the overlap of different genetic histories. A population with much type A but little type B will produce relatively few AB individuals. A population with both A and B in meaningful frequencies will produce more. In that sense, AB is a quiet witness to mixture, though not always recent mixture, and never in a simple one-to-one way. A person with AB does not prove a dramatic ancestral collision in the last few generations. The necessary A and B lineages may have been present together for centuries or millennia. Still, the type itself reminds us that human blood maps are not built only by separation. They are also built by reunion. The medical identity of AB is almost the reverse of type O. People with type AB red blood cells carry both A and B markers, so their immune systems usually do not produce antibodies against either A or B. In transfusion terms, this makes AB plasma especially valuable in certain contexts, while AB red cells are more limited as donations because they carry both markers. Again, modern medicine reveals the importance of a system that ancient people never named. In deep history, the evolutionary meaning of AB must have involved the same older forces: inheritance, disease, reproduction, drift, and population contact. Because AB requires both A and B, it is often most visible where the distributions of A and B overlap. Parts of Eurasia, where long histories of migration and mixture brought diverse populations into contact, provide many such settings. Trade routes, farming frontiers, steppe movements, imperial expansions, urban centres and marriage networks could all help sustain the conditions in which AB appears. But its rarity persists because even in mixed populations, inheriting A from one side and B from the other is only one possible outcome among many. O remains common in many places, and A or B may dominate locally. A B is therefore both a product of combination and a reminder that combination has probabilities, not guarantees. There is a tempting story that AB must be a very recent blood type because it appears less often and depends on A and B meeting. The reality is more cautious. AB individuals could have existed whenever and wherever A and B alleles coexisted in the same breeding population. That could be ancient in some places. What may be more recent are some of the distributions and frequencies we see today, shaped by historical movements, expansions, and mixing. The evidence remains incomplete, especially because ancient DNA studies are still building the long timeline of blood group frequencies. It is safer to say that AB often reflects overlap and mixture, rather than to assign it one dramatic birth date. AB also illustrates why blood type cannot be used to define identity. If one blood type already resists simple interpretation, AB resists it even more. It does not belong to one continent, one culture, one class, one ancient lifestyle, or one destiny. It is created by inheritance from both sides of the ABO system. Its carriers may descend from populations that have mixed recently, or from communities where A and B have long coexisted. Its rarity makes it interesting, but rarity is not the same as supernatural significance. Popular myths often gather around rare traits because rarity feels meaningful. Evolution, however, often produces rarity through ordinary mathematics. There may also be health associations connected to AB as with other blood types, but they are complex and should not be treated as personal predictions. Some studies have explored links between ABO type and risks for clotting, cardiovascular conditions, certain infections, or immune responses. A B sometimes appears in these discussions because it carries both A and B markers, but the size and meaning of such associations vary. What survives in the record suggests that blood type can influence biological tendencies in small ways, yet lifestyle, environment, ancestry, medical care, and chance usually matter enormously. AB does not make a person fragile or powerful by itself. It is a marker, not a fate. In ancient life, AB would have been invisible. No one in a Bronze Age town, a steppe camp, a river valley village, or a medieval port would know that a child carried both A and B markers. The child would be judged by strength, kinship, labour, beauty, skill, obedience, courage, or luck, not by blood type. Yet the blood marker would still sit on cell surfaces, participating in the unseen conversation between body and world. If certain pathogens interacted differently with A or B markers, AB might have carried its own blend of risks and protections. Scholars still debate the details, and no single explanation fully accounts for AB's persistence. Its clearest story remains demographic. It is the child of overlap. That overlap is one of the great themes of human history. Early human groups separated across landscapes, but they also met again. Ice retreated, rivers opened, deserts shifted, boats crossed sea gaps, herders entered farming zones, traders built routes, armies broke borders, and captives were taken into new households. Genetic histories that had once moved apart came back together. The ABO system records some of this in miniature. A and B lineages meeting in one body produce AB, a tiny biological trace of the fact that humanity is not a set of sealed containers. It is a long history of separation and return. There is something almost poetic in that, though the science itself needs no poetry to be meaningful. AB reminds us that rarity can emerge from connection. It is not rare because it is isolated from the human story, but because it depends on particular combinations within it. It appears where ancestral lines cross, where probabilities align, where a child inherits two different molecular instructions and carries both. Its existence quietly undermines any attempt to turn blood type into rigid categories of people. The rarest familiar type is by definition a union. By this point, the ABO story has carried us through ancient primate inheritances, human migrations, disease, pressures, farming worlds, and mixed populations. But ABO is only the most famous blood group system, not the only one that matters. Another marker lies just beyond it, medically crucial, and surrounded by even more misunderstanding. It has inspired real science, dangerous myths, pregnancy tragedies, and modern life-saving treatments. It is called the Rh factor, and its story begins with a simple positive or negative sign that conceals one of the most haunting complications in human blood. The Rh factor enters the blood story with a deceptively small sign, positive or negative, a mark on a medical form, a detail checked before transfusion, pregnancy care, surgery, or emergency treatment. It looks almost too simple beside the older alphabet of ABA, B, and O. Yet behind that sign is another vast system of inherited markers, medically powerful and evolutionarily puzzling. If ABO blood types reveal one layer of human history, Rh reveals another, and it has drawn around itself a fog of fascination because its distribution is uneven, its consequences can be severe, and one form of it, Rh-negative blood, is unusually common in some populations and rare in many others. The name itself comes from early research connected to rhesus monkeys, though the human Rh system is not simply monkey blood copied into human bodies. Modern science recognizes the Rh blood group as a complex system involving several antigens, with the D antigen being the one most people mean when they say Rh positive or Rh negative. If a person has the D antigen on their red blood cells, they are usually called Rh positive. If they lack it, they are called Rh negative. This absence can matter enormously when Rh negative blood encounters Rh positive blood, because the immune system may recognize the D antigen as foreign and produce antibodies against it. Like ABO Rh status is invisible without testing. A person does not look Rh positive or Rh negative. No ancient community could have sorted people this way by sight, and yet the marker could affect life and death, especially in childbirth. That is one reason Rh negative blood has become surrounded by stories. Some of those stories are scientific, cautious, and grounded in inheritance. Others are speculative, exaggerated, or completely unsupported. Popular myths have tried to link Rh negative blood to lost civilizations, alien ancestry, supernatural traits, or secret elite lineages. These claims are not supported by mainstream genetics. They survive because rarity invites imagination, and because people often want blood to mean more than biology can honestly say. The real mystery is already powerful enough. Rh negative blood is found at higher frequencies in parts of Europe, especially among some populations of Western Europe, while it is much rarer in many East Asian, indigenous, American and African populations. That uneven distribution raises genuine evolutionary questions. Why should the absence of a blood cell antigen become relatively common in some places and remain rare elsewhere? Was it shaped by genetic drift founder effects, ancient population structure, selection, or some combination of all of them? The evidence remains incomplete, and scholars still debate how best to explain it. One likely part of the answer is demographic history. A trait can become common in a population if it is present among founders and then amplified by isolation or expansion. Europe's ancient population history involved repeated layers, Ice Age hunter-gatherers, early farmers from Anatolia, and nearby regions, steppe-associated pastoralists, and later migrations and mixtures. Rh negative frequencies may reflect some combination of these deep histories, though the details are still being refined by ancient DNA research. No single explanation fully accounts for the pattern, and it would be misleading to treat Rh negative blood as the signature of one ancient people. Another possibility is selection, but here the story is difficult. Rh incompatibility can create a serious reproductive problem when an Rh negative mother carries an Rh positive fetus and becomes sensitized to the D antigen. In later pregnancies, her antibodies can attack the red blood cells of an Rh positive fetus, causing hemolytic disease of the newborn. Before modern medicine, this could lead to miscarriage, stillbirth, severe newborn illness, or death. At first glance, this seems like a reason Rh negative blood should be selected against not become common anywhere. That contradiction is part of the enigma. If Rh negativity can create reproductive risk, why does it persist at notable frequencies in some populations? Several answers are possible, none complete on its own. Rh negative individuals are not always at risk. The danger depends on mating patterns, fetal, Rh status, sensitization, and pregnancy sequence. An Rh negative mother carrying an Rh negative fetus faces no Rhd incompatibility. Sensitization often becomes more dangerous after a first exposure, meaning the first Rh positive child may be born healthy, while later pregnancies carry higher risk. In small or isolated populations with higher Rh negative frequency, Rh negative mothers may more often have Rh negative partners, reducing the problem. There may also have been unknown advantages linked to Rh variation, or the frequency may have risen largely through drift despite its costs. What survives in the record suggests a balance of chance population structure and perhaps pressures not yet fully understood. The Rh story also reminds us that evolution does not remove every harmful trait. A genetic variant can persist if its costs are limited, context-dependent, recessive, late-acting, or balanced by other factors. Many human traits remain in populations despite risks. Some are protected by advantages in other settings. Some survive because natural selection is weaker than demographic accident. Some persist because human mating patterns and social structures reshape the odds. Rh-negative blood may be medically dramatic, but its survival does not require a hidden cosmic explanation. Ordinary evolutionary processes can produce extraordinary patterns. The medical consequences, however, are anything but ordinary for the families affected. Before Rh immunoglobulin and modern prenatal care, repeated pregnancy loss or newborn death could haunt a family without explanation. A woman might bear one healthy child and then lose later infants, while no one understood that her immune system had learned to attack a marker inherited from the father. The tragedy would appear as curse, fate, weakness, divine punishment, or simple grief. Blood would be involved, but not in the symbolic way people imagined. The danger came from an invisible incompatibility between inherited markers. This is where Rh becomes one of the most intimate blood group stories. A BO shapes transfusion and may connect to disease pressures, but Rh incompatibility enters the quiet space of pregnancy, where one body shelters another that is genetically related and yet not identical. Human reproduction depends on tolerance. The mother's immune system must protect her from threats while not destroying the developing child. Rh incompatibility exposes how delicate that balance can be. Evolution built a system that usually works, but when it fails, the result can be devastating. At the population level, Rh status is a marker of ancestry only in a limited and statistical sense. It can help describe broad patterns, but it cannot identify a person's origin with precision. An Rh-negative person may have ancestry from many places. An Rh-positive person may belong to any population on Earth. The presence or absence of the D antigen does not reveal purity, destiny, personality, or hidden power. It is one inherited feature among thousands. The myths around Rh-negative blood often arise from turning a real distribution pattern into a fantasy of exceptionalism. Science is more restrained and more interesting. It asks how such a pattern emerged without pretending the answer is simple. Rh also shows why blood type history must be told alongside medicine. For most of human existence, these markers acted without being understood. Then in the modern period, discovery transformed fate. Once doctors learned to identify Rh incompatibility and prevent sensitization, a quiet, ancient danger became manageable. A blood group that once brought unexplained loss could be recognized, monitored, and treated. The marker did not change. Human knowledge did. The Rh factor then is both an evolutionary clue and a moral reminder. It shows that biology can carry old risks into the present, but also that understanding can soften them. It separates real mystery from unsupported myth. It points toward ancient population histories still being reconstructed from bones, genes, and living distributions. And it leads directly into one of the most haunting consequences of blood type variation, the moment when the invisible inheritance of parents becomes a hidden danger for the child not yet born. The danger of Rh incompatibility begins in one of the safest places a human life is supposed to have, the womb. It does not begin with a visible wound, a contaminated river, a mosquito bite, or the crowded air of a city. It begins with inheritance. A mother carries one blood marker pattern, a fetus carries another, and the two are close enough to share oxygen, nutrients, warmth, and risk, but different enough for the immune system to notice. For most pregnancies, the boundary between mother and child is astonishingly well managed. The placenta is not a simple wall, but a living interface negotiating between two related bodies that are never completely the same. Rh incompatibility shows what can happen when that negotiation turns dangerous. The classic problem involves an Rh-negative mother and an Rh-positive fetus. The fetus may inherit the Rhd antigen from an Rh-positive father. During pregnancy, or especially during birth, small amounts of fetal blood can enter the mother's circulation. If the mother's immune system recognizes the D antigen as foreign, it can begin producing antibodies. The first exposure may not cause severe harm to the first child, because the immune response can take time to develop. But once the mother is sensitized later, pregnancies with Rh-positive fetuses may face serious danger. Maternal antibodies can cross the placenta and attack fetal red blood cells, causing anemia, jaundice, swelling, brain injury, miscarriage, stillbirth, or newborn death. In the modern world, this risk can often be prevented with Rh immunoglobulin, a treatment that stops sensitization before the mother's immune system builds a lasting attack. But for almost all of human history, no one had such protection. There was no blood typing, no antibody screen, no prenatal injection, no exchange transfusion for a newborn in crisis. Families saw only the pattern of loss. A first child might live, and then later babies might be born weak, yellowed, swollen, or not alive at all. The cause remained hidden beneath the language of fate. The evidence remains incomplete when looking far back into prehistory, but the biological mechanism is clear enough to imagine its ancient human consequences. In small communities, such losses would not have been abstract. They would have entered memory, rumour, and grief. A woman might be feared as unlucky. A marriage might be blamed. A lineage might seem cursed. Healers could offer herbs, prayers, bleeding amulets, or ritual protections, none of which could address the molecular conflict unfolding across the placenta. The tragedy would be intimate and repeated but invisible. No one could know that a tiny antigen on fetal red blood cells had taught the mother's immune system to respond as if the next child were an invader. This is one of the most haunting details in the history of blood type because it shows that evolution does not always produce harmony within the family. Parents and children share genes, but not all genes. Pregnancy is cooperation, but also biological tension. The fetus carries paternal inheritance that the mother's body must tolerate. Most of the time, complex immune mechanisms allow this tolerance to succeed. Yet Rh incompatibility reveals how a normal defense system can become misdirected. What protects one body from foreign cells can threaten the developing body inside it. From an evolutionary perspective, Rh incompatibility raises a difficult question. If Rh negative status can create such reproductive danger in certain pairings, why has it not disappeared? The answer lies in the fact that the danger is conditional. An Rh negative woman with an Rh negative partner will not have Rh positive fetuses from that pairing. An Rh negative mother may have an Rh positive first child who survives before sensitization becomes a serious issue. Sensitization is not guaranteed in every pregnancy. The severity varies. In populations where Rh negative status is more common, Rh negative mothers are more likely to have Rh negative partners, reducing the frequency of dangerous pairings. These conditions soften the force of selection. No single explanation fully accounts for the persistence of Rh negativity, but the risk is not constant enough to erase it everywhere. The story becomes even more complex when human social life is considered. Marriage patterns, kinship rules, isolation, migration, and community size all influence which blood types meet. In a small valley where Rh negative frequency is high, the risk pattern differs from that of a port city where people from many regions intermarry. In a community shaped by endogamy, certain blood markers may remain common within the group. In a frontier zone where populations mix rapidly, incompatible pairings may become more frequent. Biology does not act in an empty space. It acts through households, customs, journeys, and accidents of history. A bioincompatibility can also affect pregnancy, though usually in a different and often milder way. A mother with type O, for example, may have antibodies that react against A or B markers in a fetus. This can sometimes cause newborn jaundice or anemia. In many cases, the effects are manageable or limited, and ABO incompatibility can even reduce the chance of Rh sensitization in some circumstances, because fetal cells may be cleared before the mother mounts a strong Rh response. The interactions are complicated, and scholars still debate the evolutionary implications. What matters for the larger story is that blood type is not only about disease outside the body, it can shape the hidden risks of reproduction itself. Before modern medicine, infant and maternal mortality were already high for many reasons: infection-obstructed labor, hemorrhage, malnutrition, prematurity and poor sanitation. Rh disease would have been one danger among many and often impossible to distinguish. This makes it difficult for archaeologists or historians to identify directly in ancient remains. A newborn grave cannot easily reveal whether incompatible blood markers cause death. A pattern of infant loss in a family may leave no record at all. The absence of evidence is not evidence of absence. It is a reminder that many of the most important human tragedies leave almost no archaeological trace. There is a quiet lesson here about what history preserves. Wars leave weapons, kingdoms leave walls, elites leave tombs. But repeated pregnancy loss in ordinary families often leaves nothing that can be read centuries later. The blood story forces attention toward experiences that rarely enter chronicles. Mothers waiting through dangerous months, infants who lived only briefly, households reshaped by grief and biological conflicts no one had the tools to understand. Human evolution is not only written in grand migrations, it is written in births that succeeded and births that did not. Modern treatment transforms this ancient risk into a largely preventable condition in many parts of the world, though access to care remains uneven. A simple injection at the right time can prevent sensitization and protect future pregnancies. This is one of medicine's quiet triumphs, not dramatic in appearance but profound in consequence. It does not erase the evolutionary history of Rh variation. It changes what that history means for families now. A marker that once carried hidden danger can be managed because humans learned to see what was invisible. The shift from mystery to prevention also separates science from myth. Rh-negative blood does not imply alien ancestry, lost royal descent, supernatural sensitivity, or any of the unsupported claims that circulate around rare traits. Its real power lies in the way it exposes the hidden negotiations of reproduction and the uneven patterns of ancestry. It is mysterious because biology is intricate, not because it requires fantasy. The evidence remains incomplete in reconstructing its ancient spread, but the medical reality is firmly grounded. Blood birth and hidden risk bring the story back to a central truth: inherited markers matter most when they meet the world. Sometimes that world is a swamp full of mosquitoes. Sometimes it is a crowded village well. Sometimes it is a trade port where new diseases arrive by ship. And sometimes it is the body of a mother carrying a child with a different inheritance. Blood type is not destiny, but it is never isolated. It becomes meaningful through contact. Across generations, these contacts shape families, populations, and maps. Rh markers pass through marriages. ABO types move through migrations. Communities expand, collapse, mix, and isolate. The next layer of the story is not only biological but historical, because once humans build empires, trade networks, colonies, and routes of forced movement, blood markers begin traveling faster and farther than ever before, carried inside bodies, crossing the borders that history keeps redrawing. Once blood types enter the age of empires, they stop moving only at the pace of wandering families and small migrating bands. They begin moving along roads deliberately cut through mountains, across sea lanes, watched by merchants through military frontiers, plantation systems, slave markets, pilgrimage routes, marriage alliances, colonial ports and cities swollen with strangers. Human blood has always travelled, but civilization gives it new engines. A gene that once passed from one valley into the next over centuries can now cross a sea, in the body of a sailor, a captive, a soldier, a trader, a priest, a bride or a child taken from home and raised under another sky. This does not mean empires create blood types. The markers are far older. ABO, ABRH, positive Rh negative, and many lesser-known blood group systems already exist before the first palace wall rises. But empires rearrange the carriers. They turn local inheritance into continental mixture. They pull people from distant ecologies into shared streets and households. They create new contact zones where blood markers that once remained mostly separate begin appearing in the same family lines. What survives in the record suggests that much of the modern blood map cannot be understood through prehistory alone. It also carries the imprint of history's violent and ordinary movements. The ancient Near East offers an early example of this mixing world. River valleys draw farmers-herders, merchants and migrants into dense corridors of exchange. Mesopotamian cities depend on grain labour, irrigation and trade. Copper, tin, timber, stone, wool, and people move through networks that stretch far beyond any one city-state. Armies deport populations. Kings resettle workers. Merchants marry into foreign communities. Captives become servants, wives, soldiers and descendants. No one records blood type, but every movement changes the pool of inheritance. The city is not only a political invention, it is a genetic meeting place. Across the Mediterranean the pattern expands. Phoenician sailors establish ports. Greeks colonize coasts. Romans build roads, recruit soldiers from distant provinces, enslave conquered peoples, and move administrators, merchants and families across an empire that touches Europe, North Africa and Western Asia. In such a world, blood markers travel through both privilege and suffering. A soldier from one frontier dies on another. A slave from the East is sold in Italy. A merchant family keeps connections across the sea for generations. A child born in a port city may carry ancestry from several shores. The evidence remains incomplete for blood type frequencies in these ancient populations, but ancient DNA increasingly confirms what history already suggests: the great cities of antiquity were often more mixed than nationalist myths later imagined. The Silk Roads deepen this movement across Eurasia. These routes are not one road, but a web of passages through oases, towns, deserts, mountains, and steppe corridors. Silk horses, paper, spices, metals, religions, and diseases move through them, but so do people. Sogdian merchants, Turkic groups, Persian communities, Chinese envoys, Buddhist monks, Islamic scholars, Mongol armies, and countless unnamed families carry genetic histories across immense distances. Type B, already unevenly visible across parts of Asia, may be redistributed through some of these movements, but it is never alone. Type AO and RH variants move with the same human currents. No single blood type belongs to the Silk Roads. The routes belong to mixture. Then there are the steppe empires whose speed unsettles the settled world. Horse pastoralists can expand with astonishing reach, forming confederations that rise, fracture and reform. Their movements do not simply replace one population with another everywhere they go, but they can leave deep marks through conquest, alliance, elite dominance, marriage and migration. The genetic influence of steppe populations varies across regions, strong in some places and lighter in others. Blood type frequencies may shift as part of that broader ancestry, though they cannot by themselves identify the movements. The temptation to attach type B or any other marker to a single steppe people is too simple. What survives is a blur of expansions, absorptions, and local survival. In Africa, blood markers move through equally complex histories. The Bantu expansions spread languages, farming, ironworking, and ancestry across much of sub-Saharan Africa over millennia, interacting with older hunter-gatherer and pastoralist populations. Trans-Saharan trade links, West Africa, North Africa, and the Mediterranean. The Indian Ocean connects East African coastlines with Arabia, Persia, India, and beyond. Later, the Atlantic slave trade forcibly moves millions of Africans into the Americas, one of the most devastating human displacements in history. These movements do not merely alter culture and demography. They carry blood group frequencies into new continents, where they mix under conditions shaped by violence, survival, and resistance. The Americas after 1492 become one of history's most dramatic examples of blood map disruption. Indigenous populations, many with high frequencies of type O face, introduced diseases, warfare, dispossession, forced labour famine, and social collapse. Europeans arrive with their own varied blood type patterns. Enslaved Africans are transported in vast numbers carrying diverse West and Central African ancestries. Later migrations from Asia, the Middle East and other regions add further layers. The modern blood type distribution of the Americas is therefore not simply a preserved prehistoric pattern. It is a record of ancient founding colonial catastrophe-forced movement, survival, and mixture. No single explanation fully accounts for it, and any honest account must hold both biology and violence in view. Colonial empires accelerate these processes globally. Ships link continents with regularity. Port cities become laboratories of ancestry, though that phrase must not make the suffering sound clean. Sailors die far from home. Indigenous people are displaced. Enslaved people are renamed and sold. Convicts are transported. Labourers are recruited, coerced, or trapped by debt. Soldiers are stationed overseas. Missionaries and administrators create households across colonial boundaries. Blood markers travel through all of it. Some journeys are chosen, many are not. The genes remember movement without recording consent. This is one reason blood type should never be romanticized too easily. It can reveal contact, but contact is not always peaceful. It can reveal mixture, but mixture is not always equal. A modern person's blood type may descend from love, migration, conquest, enslavement, adoption, refuge, or survival after catastrophe. The marker itself does not tell which. It is only a molecular remnant of human events whose emotional truth may be lost. Scholars still debate how to connect genetic patterns to specific histories without oversimplifying them, and that caution is necessary. Blood can show that people moved. It cannot always tell whether they moved freely. At the same time, the movement of blood through empires undermines ideas of human purity. Every empire imagines boundaries, but empires also break them. They classify people, tax them, rank them, enslave them, marry them, deport them, and employ them across distance. The result is constant mixing beneath the language of separation. Even societies obsessed with lineage often contain hidden complexity. Blood type frequencies remind us that human populations are historical processes, not sealed containers. The borders on a map are young compared with the inheritance inside bodies. The rise of modern science brings another change. Once blood can be typed, these old movements become partly visible. Doctors and anthropologists begin collecting blood group data, sometimes with genuine medical purpose, sometimes through the distorted lens of racial classification common in earlier eras. Some researchers try to use blood types to define human groups too rigidly. Later genetics shows how flawed such simplifications are. Blood type frequencies can help describe population history, but they are not enough to divide humanity into fixed biological races. Human variation is real, patterned and meaningful, but it is gradual, overlapping and shaped by constant movement. The deeper lesson is that empires do not erase evolution, they become part of it. Culture power, violence, trade and law change who meets whom, who has children, who survives and who is displaced. Blood group markers, ancient as they are, continue to be reshuffled by human institutions. The bloodstream becomes a place where prehistory and recorded history touch. A marker shaped perhaps by ice age drift or ancient disease can be carried into a colonial port mixed through generations, and appear today in a child whose ancestry spans continents. So the map grows denser. Type O still carries echoes of founder effects and disease landscapes. Type B still follows uneven Eurasian trails. Type A still moves through old farming and urban histories. AB appears wherever A and B meet. RH negative persists with its own unresolved pattern. But now these markers are no longer only clues from the deep past. They are also witnesses to empire migration, captivity trade, and the restless human habit of crossing every boundary eventually. To see beyond the living map, however, science has had to open another archive. Not the blood of the living, but the bones of the dead. Ancient DNA recovered from teeth and skeletons is beginning to show which blood group variants existed in people who walked through vanished worlds. It does not answer everything. The record is fragile, patchy, and biased toward places where DNA survives. But for the first time, the old blood story can be read not only from modern descendants, but from the ancient bodies themselves. For most of the history of blood type research, scientists had to look at living people and reason backward. They measured blood group frequencies in modern populations, drew maps, compared regions, and tried to reconstruct the paths that might have produced them. This was useful, but it had a serious weakness. Living populations are the survivors of everything that has happened before migrations, collapses, plagues, conquests, replacements, marriages, bottlenecks, and recent expansions. To look only at the living is to read the last page of a manuscript and guess how all the earlier pages were written. Ancient DNA begins to change that. It does not make the story simple, but it allows the dead themselves to answer some of the questions. The archive is hidden in bone and teeth. Under the right conditions, fragments of DNA can survive for thousands or even tens of thousands of years. Cold caves, dry environments, and protected burial contexts can preserve genetic material long after soft tissue has vanished. Teeth are especially valuable because the hard enamel and inner structures can protect DNA from decay and contamination. From these fragments, researchers can sometimes reconstruct parts of a person's genome, including genetic variants linked to blood group systems. A skeleton no longer has flowing blood, but it may still preserve the instructions that once shaped that blood. This is a remarkable turn in the human story. A person buried in a Neolithic grave, a Bronze Age steppe burial, an Ice Age cave, or a medieval cemetery may carry genetic clues that reveal ancestry, kinship, disease, diet, and sometimes blood group markers. The dead no longer speak in language, but their cells have left a damaged biological record. What survives in the record suggests that ancient populations were often more dynamic than once believed. Farming expansions, steppe migrations, and repeated episodes of mixture can now be traced with a clarity that older archaeology alone could not always provide. For blood types, ancient DNA is still an emerging and imperfect tool. Not every ancient genome has enough coverage to identify ABO or RH variants confidently. DNA preservation varies sharply by climate and soil. Hot, wet environments often destroy genetic material, which means the ancient DNA record is biased toward colder or drier regions. Many parts of Africa, South Asia, Southeast Asia, and the tropics remain underrepresented, not because their histories are less important, but because DNA survives less easily or has been less extensively sampled. The evidence remains incomplete, and any map built from ancient DNA must be treated as provisional. Even with these limits, ancient DNA is transforming the blood type question by adding time depth. Instead of asking only why type A, BO, or Rh negative appears in modern populations, scientists can ask when certain variants appear in ancient remains and how their frequencies change across periods. Did a blood group become more common after farming spread? Did it shift after steppe ancestry arrived? Did a rare marker exist in a region before a known migration, or did it appear afterward? These questions cannot always be answered yet, but they are now answerable in principle. The method also helps separate myth from history. Claims about pure ancient bloodlines often collapse when ancient DNA is examined. Populations once imagined as isolated or unchanged frequently turn out to be mixtures of earlier groups. Europe, for example, is not the product of one timeless population but of repeated layers. Ice Age hunter-gatherers, early farmers, steppe pastoralists, and later historical movements. The same is true in different ways across much of the world. Blood type frequencies sit within these broader genetic transformations. A marker's presence in modern people may reflect many layers, not one ancestral essence. Ancient DNA can also reveal lost diversity. Some lineages that once existed have little or no direct continuation today. Others were absorbed into expanding populations. If certain blood group variants were present in groups that later declined or mixed, their traces may survive faintly or vanish almost entirely. This matters because modern frequencies can hide the past as much as reveal it. A blood type rare today in a region may once have been more common. A type common today may have risen only after a later expansion. Without ancient samples, those shifts remain invisible. There is a haunting intimacy to this work. A tooth from a long-dead child may contain not only ancestry, but clues to inherited blood markers. A warrior burial may reveal that the person's genetic background came from far beyond the place of burial. A farmer interred near a village wall may carry ancestry from migrants who arrived generations earlier. These are not just data points. They were bodies that once warmed beside fires, suffered infections, carried children, crossed landscapes, and lived inside social worlds now almost entirely gone. Blood group genetics brings their biology into the present without restoring their voices. The study of ancient pathogens adds another dimension. In some remains, researchers can identify DNA from microbes that infected people at or near death. Plague bacteria, hepatitis viruses, tuberculosis-related organisms, and other pathogens have been recovered from ancient remains in some cases. When ancient human genomes and ancient pathogen genomes are studied together, a deeper question becomes possible. How did disease pressures interact with human genetic variation over time? Blood groups may one day be part of that larger reconstruction. Scholars still debate many details, but the direction is clear. The history of blood is also the history of the organisms that tried to use human bodies as habitat. This does not mean every ancient epidemic will be matched neatly to a blood type shift. The past is too messy for that. A plague may devastate a city without leaving a clear blood group signature. A migration may change blood frequencies more dramatically than disease. A local founder effect may mimic selection. Ancient DNA can sharpen the picture, but it cannot remove all uncertainty. It is still a record of fragments sampled unevenly, interpreted through models that improve over time. No single explanation fully accounts for the living blood map even with ancient DNA. Yet the power of ancient DNA is that it restores change. Modern people often imagine ancestry as stable, but ancient genomes show movement everywhere. The old world was not static. Hunter-gatherer territories shifted. Farmers expanded, pastoralists moved, island populations formed and reformed. Cities drew people from distant regions. Epidemics struck and reshaped communities. Blood group variants moved through all of this, sometimes rising, sometimes falling, sometimes simply enduring. The bloodstream becomes less like a fixed identity and more like a current fed by many tributaries. There is also a caution. Ancient DNA is powerful, but it can be misused when people try to force it into simple stories of belonging or superiority. Blood type data is especially vulnerable to this because blood already carries symbolic weight in human culture. The language of blood has been used to define family, nation, class, race, and exclusion. Modern genetics shows that such boundaries are far more porous than those ideologies admit. The more ancient DNA reveals, the harder it becomes to believe in pure lines. The dead themselves testify to mixture. As the archive grows, the story of blood type will likely become more detailed and more surprising. Some assumptions may be confirmed, others may fall away. Researchers may discover that certain blood group frequencies shifted at unexpected times, or that ancient populations carried combinations rarely seen today. The evidence remains incomplete, but every new genome adds another small piece to the map. A tooth, a petrous bone, a grave beneath a city, a skeleton from a drowned landscape, each may preserve a trace of the invisible markers that once moved through living veins. Ancient DNA does not close the mystery. It changes its shape. It gives the blood story a timeline, but the timeline is still broken. It reveals movement, but not always motive. It shows ancestry, but not the full human meaning of migration, marriage, exile, or survival. It can identify a genetic variant, but it cannot tell whether the person who carried it died, afraid, loved deeply, or watched the seasons with patience. Science can recover the code. It must remain humble before the life. And that humility matters as the story approaches its most important limit. Blood type can reveal real things about human evolution, migration, population, structure, disease, pressure, reproductive risk, and ancient mixture. But it cannot reveal everything. It cannot carry the burden people sometimes place on it. It cannot explain personality, destiny, moral worth, or the full truth of ancestry. To understand what blood type reveals, we must also understand what it does not. Blood type becomes dangerous when people ask it to explain too much. A small set of inherited markers can tell real stories about human evolution, but it cannot carry the weight of identity, destiny, personality, or worth. It can help trace population patterns, suggest old migrations, illuminate medical risks, and hint at the long pressure of disease. But it cannot reveal whether someone is brave or gentle, ancient or modern, superior or weak. The trouble begins when a biological clue is treated as a prophecy. Human beings have always wanted blood to mean more than blood can honestly mean. Across many cultures, blood has been used as a language of belonging. People speak of bloodlines, royal blood, mixed blood, pure blood, blood, guilt, blood, debt, blood, brotherhood, and blood inheritance. These ideas existed long before anyone understood ABO or RH systems. They are emotional, social, political, and symbolic. When modern blood typing appeared, it entered a world already prepared to overinterpret it. A laboratory result could become in the public imagination a sign of hidden nature. That temptation has never fully disappeared. Some popular traditions have assigned personality traits to blood types, claiming that type A people are orderly, type B people are creative, type O. People are confident, and type AB people are unusual or complex. These claims may be culturally influential in some places, but they are not supported as reliable science. They turn broad biological categories into personal mythology. The evidence remains incomplete for many subtle biological effects of blood type, but personality is not one of the things ABO can meaningfully reveal. A person's temperament emerges from genes, development, family culture, experience, health, memory, and chance. A surface marker on red blood cells cannot explain the architecture of a human mind. There are also myths around diet. Some claim that each blood type corresponds to an ancient eating style, and that people should eat according to whether their blood supposedly descends from hunters, farmers, or nomads. These stories sound satisfying because they turn evolution into a menu, but they oversimplify both blood history and human diet. Foragers ate many different diets depending on ecology. Farmers were not one biological type. Pastoralists, fishers, gardeners, city dwellers, and forest peoples all carried mixed blood group patterns. Human nutrition is shaped by metabolism, environment, culture, gut microbes, health conditions, and food access, not by ABO alone. What survives in the scientific record does not support blood type as a master guide to diet. The same caution applies to ancestry. Blood type frequencies can describe populations statistically, but they are blunt tools for identifying individual origins. A person with type O may have ancestry from almost anywhere. A person with type B is not automatically linked to one migration or region. Rh-negative status does not prove descent from a mysterious ancient people. AB does not prove recent exotic mixture. Blood types are shared across huge numbers of unrelated people because the variants are old and have moved repeatedly through history. They can contribute one small piece to population genetics, but they cannot replace genome-wide ancestry, archaeology language, or documented family history. This limit is important because blood has often been misused in racial thinking. Earlier researchers sometimes tried to classify humanity too rigidly by blood group frequencies as though populations were fixed biological boxes. Modern genetics has undermined that view. Human variation is real, but it is mostly gradual overlapping and shaped by repeated mixture. There are no clean biological walls separating human groups. Blood type patterns can vary by region, but every region contains diversity, and most blood group systems cut across cultural and national boundaries. No single marker can define a people. The deeper problem is that blood feels intimate. It seems to come from the core of the body, and because it is inherited, people imagine it as a direct line to the past. In one sense, that feeling is not wrong. Blood group genes do connect us to ancestors, but they connect us in a narrow way. Each person inherits only particular variants among millions of genetic positions. A blood type is one thread in an enormous fabric. To hold up that thread and claim it explains the whole tapestry is to misunderstand the scale of inheritance. Even medical associations require care. Studies may find that certain blood types have slightly different risks for clotting, some infections, ulcers, or other conditions. These findings can matter for research and public health, but they're usually not personal verdicts. Risk is not certainty. A small statistical association does not mean an individual will develop a disease or avoid one. Age, environment, vaccination, sanitation, diet, exercise, medical care, genetics, beyond blood type and simple chance all matter. Blood type belongs in medicine, but it should not become fatalism. This is especially true when people hear that one blood type may have an advantage against one disease. Such claims easily become distorted into the idea that a blood type is stronger. Evolution does not create strength in the abstract. It creates trade-offs. Type O may be associated with some protection against severe malaria in certain contexts, but also with greater vulnerability to severe cholera. Rh-negative status can be ordinary in daily life and dangerous in certain pregnancies without treatment. Type A or B may carry different associations depending on the disease being studied. No blood type is universally best because there is no universal environment. The limits of blood type interpretation do not make the subject less fascinating. They make it more honest. The real story is not that blood reveals who someone truly is. The real story is that blood reveals how much of human life has been shaped by pressures no one could see. It shows that survival often depended on small differences interacting with vast circumstances. It shows that migration and disease can leave patterns in the body. It shows that modern medicine can turn ancient risks into manageable facts. It shows that the human past is present inside us, but not in a way that flatters simple myths. There is a kind of humility in this. A blood type can be ancient without being mystical. It can be meaningful without being decisive. It can be medically important without being a personality map. It can carry traces of ancestry without defining identity. What survives in the record suggests that blood groups are best understood as clues, not conclusions. They are fragments of a much larger human archive. So when a person learns their blood type, they are not discovering a secret destiny. They are seeing one small sign of a long evolutionary history shared with countless strangers. Someone on another continent speaking another language, living another kind of life may carry the same marker. Someone in an ancient grave may have carried it too. The connection is real, but it is not exclusive. It belongs to humanity's tangled inheritance. Blood cannot tell us everything. It cannot restore vanished voices, explain every migration, identify every ancestor, or solve every mystery of disease. But within its limits it does something extraordinary. It reminds us that the body is historical. Every cell exists in the present, yet carries structures shaped by the past. To understand blood type properly is to resist both emptiness and exaggeration. It means seeing the marker for what it is, a small, durable trace of evolution, moving through a species that has never stopped changing. And once the myths are set aside, the final image becomes clearer. Blood type is not a horoscope, not a rank, not a tribal seal, not a hidden verdict. It is a living map of survival, incomplete, but real, drawn through migration, disease, birth chance, and time. The last part of the story is not about what blood says about one person alone, but what it says about all of us as descendants of survivors, walking forward with ancient markers still moving quietly beneath the skin. A living map of survival does not look like a map at first. It looks like an ordinary person standing in ordinary light, unaware that their blood carries traces of vanished climates, ancient infections, crowded villages, long migrations, narrow escapes and births that shaped the future one generation at a time. The markers are silent. They do not announce themselves in the face or voice. They do not preserve a complete ancestry or reveal a hidden destiny. Yet they remain inside the body as small, durable signs of a human story far older than written memory. By the time blood type reaches the present, it has passed through nearly every force that makes humanity difficult to summarize. It has moved with Ice Age foragers and early farmers, with pastoralists crossing open grasslands, with sailors following stars and coastlines, with captives forced across oceans, with migrants seeking land work, safety or escape. It has been shaped by chance in small founding groups and by disease in crowded settlements. It has been protected, punished, ignored and rearranged by environments that changed faster than bodies could fully adapt. What survives in the record suggests that blood type is not one story, but many stories overlapping inside the same vein. The most important lesson is not that one blood type is older, stronger or more mysterious than another. The lesson is that human survival has always depended on variation. A species too uniform is fragile when the world changes. Diversity in immune markers, blood groups, metabolism, and countless other traits gives populations many ways to endure uncertain conditions. Some differences matter greatly in one age and barely at all in another. Some become useful only when a particular disease appears. Some persist because they do little harm. Some survive through pure accident. No single explanation fully accounts for the pattern, because the pattern was made by life itself. The blood type written on a medical form is therefore both practical and ancient. It can guide transfusion, it can protect pregnancy, it can help doctors avoid disaster. But behind that modern use is a deeper inheritance shaped long before anyone knew what blood groups were. The same marker that matters in a hospital today may once have moved through a body fighting fever, beside a river crossing a mountain pass surviving famine, or giving birth in a house of mud smoke and fear. The connection is not direct enough to turn into legend, but it is real enough to make the present feel older. In the end, blood type reveals something quietly profound about human evolution. It shows that the past is not only buried in caves, bones, tools, ruins, and ancient DNA laboratories. It is also circulating in living people. The body is not separate from history. It is one of history's archives. Every generation receives a damaged, revised, unfinished manuscript from the one before it, and then passes it on changed by survival, love-loss movement, and chance. And this is where the mystery softens rather than disappears. Blood type does not solve the human story. It reminds us how much of that story happened beneath awareness. Our ancestors did not know the names of the markers they carried. They only knew hunger, rain sickness, birth danger, and the need to keep going. Yet because some did keep going, the markers remain. They are not grand monuments. They are smaller than dust, smaller than memory, but they have crossed more time than any empire. The blood inside us is not a prophecy. It is a trace, and like all traces, it asks to be read carefully. The long journey of blood type ends for now not in a laboratory or an ancient grave, but in the quiet continuity between the dead and the living. Somewhere in the deep past, a child survived a fever. Somewhere else, a small group crossed into an empty coast and became the ancestors of many. A mother carried a pregnancy to term. A village endured a season of disease. A traveller reached a new shore. A captive survived long enough to have descendants. A farmer, a herder, a hunter, a sailor, a healer, a soldier, and a nameless child all carried markers they could never see. Those markers moved on. They passed through centuries of dust and rain, through the rise of cities, through the fall of kingdoms, through ships, roads, borders, and graves. They entered the modern world disguised as a simple line on a chart ABABO, positive negative. But behind those letters and signs is the slow pressure of time, the unfinished record of adaptation, and the fragile luck of being descended from those who endured. So the next time blood is treated as ordinary, it is worth remembering how old that ordinary thing is. It has crossed ice ages without speaking. It has survived diseases without understanding them. It has carried the marks of landscapes that no longer exist. It has outlived languages, gods, villages, and empires. And still it moves quietly beneath the skin, red and warm, keeping its ancient record in motion. Not as a secret code, not as destiny, but as a living remnant of the long human passage through danger, chance, and time.